We are going to discuss the workbook question on Chapter 1, which is Genetic Inheritance and Genetic Control. Question 1. Inflated pot shape is the dominant trait. So Irene needs to identify the genotype of a pea plant with inflated pot shape as either homozygous dominant or heterozygous. So she would do a test cross with a pea plant with genotype homozygous recessive. So the answer would be A. So in this question, you have to understand how to determine the genotype of an individual with dominant phenotype. So in this case, the dominant phenotype is inflated pot shape. Okay, so this dominant phenotype, the genotype could either be homozygous dominant or heterozygous. So you have to determine which one is the genotype. So in order to determine the genotype, you have to cross this individual that you don't know the genotype with an individual with homozygous recessive. So in this case, the genotype of homozygous recessive is small r. So if there are two outcomes, okay, so if the unknown individual is homozygous dominant, okay, capital R, you cross with homozygous recessive, small r, and then you would get the F1 generation, or the offspring is in the condition of heterozygous. Okay, so this heterozygous offspring would show or would exhibit dominant phenotype. So in the F1 generation, all of the offspring, if it shows dominant phenotype, which is inflated pot shape, so it means that the unknown individual is homozygous dominant. But if the unknown individual is heterozygous and you cross with homozygous recessive, so the F1 generation that you will get is there are two outcomes, okay, of the phenotype of F1, which is in the F1 you will get half of the population of F1 will have inflated pot shape, okay, and the other half of the F1 population will be or will exhibit constricted pot shape. So 50% inflated or it shows dominant phenotype, and another 50% it shows recessive phenotype. So it means that this unknown individual would be heterozygous. A dihybrid cross between a tall chili plant with the genotype homozygous dominant with axial flower position with homozygous dominant genotype to a dwarf chili plant homozygous recessive with a terminal flower position homozygous recessive produces F1 generation of tall plants with axial flower position. The F1 offspring are allowed to self-pollinate to produce F2 generation. So therefore you have to predict the phenotypic ratio of F2 generation. For this question, you have to understand that in the dihybrid cross, it involves two genes that control two characters. Okay, so that's why we call it as a dihybrid. So you have to extract out all the information given in the question. So in this question, it tells you that the tall phenotype is a dominant phenotype. So it is controlled by the dominant T allele. So for an individual that has homozygous dominant phenotype, capital T, and heterozygous will exhibit tall phenotype. And the recessive phenotype will be dwarf, and it is controlled by the recessive small t allele. So if an individual has homozygous recessive phenotype, which is small t, so the individual will be dwarf. The second character is flower position. So the flower position, the dominant phenotype is axial, which is controlled by the dominant A allele. So individual with homozygous dominant genotype, capital A, or heterozygous will have axial flower position, and the recessive trait or phenotype is terminal. So it is controlled by the small a allele, and an individual with homozygous recessive phenotype homozygous recessive will have terminal flower position. So after you have extract out all of this information, so you have to do the crossing. So for the parental generation, it tells you that the parental generation has tall and axial flower position. It is tall and has axial flower position. So the genotype will be capital T, okay, for tall, and terminal will be, sorry, axial flower position will be capital A. Okay, so you have to write the alleles of the same gene side by side. Okay, alleles of the gene would you have to write it side by side. So you will never, okay, do not what we call it as, write it like this. Don't ever selang-selikan allele tu. Okay, so this will be wrong. And for the second parents, okay, it has the phenotype dwarf and terminal flower position. Okay, so after you have determined the genotype of the parent, so you have to know what is the gamete, okay, for each of the parents. So for the first parent here, let's say this is the female parent, so the gametes the female parent will produce is capital A, capital T, okay, capital T, capital A. And for the second parent, let's say that this parent is a male parent, okay, so the gamete for the sperm will have the allele small t, small a, okay. So when the sperm fertilizes the egg, it gets the F1 generation. So the F1 generation is in the heterozygous condition, and it shows all of the F1 generation will have tall and axial flower position. So this F1 generation, you allow it to undergo self-pollination. So now you have to determine what are the possible combinations of allele in the F1 gametes. So these are the alleles that you will get: capital T, capital A, capital T, small t, capital A, and small t, small a. Okay, so for this genotype that has two genes, okay, with 4 different alleles, you will get 4 different gametes. Okay, 4 different gametes. So the gametes of F1 generation, you have to put it in a Punnett square. Okay, so this is the Punnett square that you will do. Okay, so let's say like this is the sperm, and this is the eggs, okay, for the F1 generation. So when the sperm fertilizes the egg, so you have to combine all the alleles together in the gametes, okay, to get the genotype of F2 generation. Okay, so after you have combined, let's say this the allele capital T, you combine, you get homozygous dominant for tall, okay, and then capital A, you will get axial flower position. So each time you write the genotype in the Punnett square for the F2 generation, make sure down below you write the phenotype. Okay, so let's say this, let's say for example this one, okay, capital T, capital A, small a. So you will get tall and axial. So you write all the phenotype for each genotype. Okay, so after that you have to count. Okay, you have to count the phenotype in the F2 generation. So you will get, after you have done after you have completed the Punnett square, you will get 9 tall axial, 3 tall terminal, 3 dwarf axial, and 1 short terminal. So if you see the phenotypic ratio of F2 generation, the 9 here, it shows dominant phenotype, okay, and 1, it shows the recessive phenotype for both characters. Okay, so the 3 tall terminal here, it produces a totally new phenotype that is different from the parental generation and also F1 generation. Same goes to the 3 dwarf axial here. Okay, it produces a totally new phenotype that is different from the parental generation and also F2 generation. Question 3. Which of the following statement is not true regarding the patterns of inheritance? So let's look at the choices. We are going to look at choices, the last choice first, which is D. Ephistasis is when the inheritance of coat color in mice involves a gene that controls the pigment deposition, which can suppress the expression of another gene as either black nor brown coat color are produced. So for ephistasis, you have to understand it involves two genes. Okay, two genes. One gene will suppress the expression of another gene. So in this case, the first gene, okay, is for example, here in this question is for pigment. It controls pigment deposition. So pigment can be produced. Okay, this gene will determine whether pigment will be produced or not. For the second gene, it controls the color. Okay, the color of the coat, the color of the coat in mice. Okay, as either black or brown. So in this case, the first gene, which is pigment deposition, will control the expression of coat color. So this is ephistasis. So the statement D is correct. For C, the inheritance pattern of human height, okay, in a population is influenced by additive effects of more than one gene and also the environmental factors. For statement C, the inheritance patterns of human height is actually a polygene. Okay, it is polygenic. One character is controlled by many genes. Okay, many genes, and each gene will contribute the additive effect to that phenotype. Okay, so height is also influenced by the environmental factors. Okay, so for example, if you take a set of twin, okay, a set of twin, obviously this twin will have the same genetic composition. So initially, the height of these two individuals of this twin will have the same height. But if you were to give one of the twin or to feed one of the twin with milk, okay, with milk, and the other one you just give juice, okay, so obviously the twin will show difference in height. Okay, so this is what we call it as polygenic inheritance. Okay, it is influenced by the gene and also environmental factors. So another example of polygenic inheritance is skin color. Okay, so skin color, the color of the skin is influenced by the expression of the gene and also environmental factors. So if you expose this set of twin, initially they have the same genetic composition, but if you expose the twin to different exposure of sunlight, okay, one you only allows the twin, one of the twin to be inside the house, the other one you allow one of the twin to play football in the hot in the hot temperature outside. Okay, so obviously they will show difference in the skin color. Okay, so the statement for C is correct. For B, in complete inheritance, the genotypic ratio and the phenotypic ratio of F2 generation is in the 1 to 2 to 1. Okay, so for incomplete dominance, for example, you cross a flower that has red color to a flower with white flower color. Okay, so for example, the genotype for red is capital R, white is small r, and the F1 generation you produce is heterozygous, capital R, small r. But okay, the phenotype will be pink. Okay, so you allow F1 generation to undergo self-fertilization. Okay, so then you get the F2 generation in the 1 to 2 to 1 phenotypic ratio. Okay, so this is the genotype. of the F2 generation. Capital R, capital R, capital R, small r, capital R, small r, small r, small r. Okay, so you will get 1 red to 2 pink to 1 white. Okay, so the phenotypic ratio is the same as the genotypic ratio for the F2 generation. So statement B is correct. For statement A, the inheritance patterns of human ABO blood group system involves multiple allele. Okay, so human blood group here, it is controlled by 3 different alleles: IA, IB, and IO. So is it only controlled by multiple allele? No, it is also controlled by codominance. For example, if an individual has the blood type AB, okay, so it means that the genotype of this individual is IA, IB, okay, IA, IB. So for individuals that has the genotype IA, IB, okay, so the red blood cells of this individual will have both antigen. Okay, antigen A, for example, this triangle here is for antigen A, and circle here is for the production of antigen B at the surface of red blood cells of that individual. Okay, so it means that both for ABO blood group, it shows codominance. Both of the alleles are equally strong, and both of the alleles will be expressed in the offspring. Figure 1 shows the inheritance patterns of attached earlobe in a family. This trait is caused by recessive alleles. Which of the following statement can be concluded from the pedigree? So for pedigree analysis, you can analyze the inheritance of traits, okay, that are controlled by dominant trait or dominant allele or recessive allele. So we have pedigree analysis for dominant trait, okay, and also pedigree analysis for recessive trait. So in this case, in this question, for example, it is actually pedigree analysis for recessive trait. So in order for you to analyze this pedigree, you have to look at if an individual has attached earlobe, so that's mean they are in the homozygous recessive condition. Okay, they are homozygous recessive. So let's say, for example, we are going to assign 2 different alleles, okay, which is capital F for free and attached earlobes small f. Okay, so for free earlobes, for free individuals that has free earlobes, the genotype could either be homozygous dominant or heterozygous. But for individuals that has attached earlobe, they are obviously just only homozygous recessive. So the genotype will be small f, small f. Okay. So that's mean if the individual has free earlobes, they could either be homozygous dominant or heterozygous. So if you look at the parents here for the parental generation, so they show free earlobes, but the parents could either be homozygous dominant or heterozygous. But if you look at the offspring, the F1 generation, the offspring of the parental generation, they will express the phenotype, okay, which is attached earlobe. So the genotype will be small f, small f. So obviously the parents has to be heterozygous. Okay, the parents has to be heterozygous. So the answer will be A. So parents that are heterozygous or heterozygous individuals, we call them as carriers, because they carry the recessive allele, but the recessive allele in the heterozygous condition are not expressed. Question 5. In a population that is in a Hardy-Weinberg equilibrium, 1 out of 700 people has a cancer caused by a recessive allele, which is small w. Calculate the proportion of individuals that carry the W allele but are not expected to have the cancer. So for this type of question, you have to understand, in the statement it says 1 out of 700 people has the cancer caused by the recessive allele. So it means that for this population, homozygous recessive individual, okay, homozygous recessive individual which has the genotype small w will have the cancer. So one out of 700 people will have the homozygous recessive genotype. Okay, so it says here in the question, you have to determine how many individuals carry the small w allele but are not expected to have the cancer. So you have to determine how many individuals that are actually heterozygous. Okay, heterozygous. So for this type of question, you have to know and apply two equations from the Hardy-Weinberg equilibrium, which is p squared plus 2pq plus q squared equals to 1. Okay, and the other one is p plus q equals to 1. So these are the two formula that you have to know and apply. So for p squared, it stands for, or it is for to determine the percentage of homozygous dominant. Okay, this is for homozygous dominant. This is to determine the percentage of heterozygous. Q squared is to determine the percentage or the frequency for homozygous recessive. Okay, so in that population is equals to 1, which is 100%. And for P plus Q is to determine the allele frequency. P is for the frequency for the dominant allele. Q is for the frequency of recessive allele. Okay, so all the alleles will equals to 1 or 100%. So in the question, it already gives you, okay, 1 out of 700 people has the cancer. So this is the proportion of individuals that are homozygous recessive. So you take from that formula, okay, q squared is for homozygous recessive. So 1 out of 700 people are homozygous recessive. So from this, you have to determine the allele frequency for the recessive allele. So you have to determine the value of Q, which is, you will get Q equals to 0.038. So you just use your calculator. So in the exam, you can take your calculator for this question. You can use your calculator. And then after you have determined the value of Q, so you use this formula, P plus Q equals to 1, to determine the P value, okay, the frequency of the dominant allele. Okay, so the frequency of the dominant allele, you will get P equals to 0.962. Okay, you just take for this type of question, you just take for 3 decimal point. Okay, and then the question wants you to determine how many individuals that are heterozygous. So from the first formula here, okay, you have to use 2pq. So 2pq, you just insert all the values, you will get 0.073. Okay, so this is the frequency for the heterozygous. So after you have you have determined the frequency for the heterozygous, so in the question it gives already the total number of the population, which is 700. So 700 you times by the frequency of the heterozygous here, which is 0.073, you will get 51. Okay, so 51 individuals out of 700 people in that population, they are heterozygous. Question 6. In the experiment performed by Michelson and Stott, the product of DNA replication after the first generation was centrifuged in a concentrated cesium chloride solution. Which of the following results would be observed after centrifugation? So the experiment carried out, these two scientists, Michelson and Stott, they carry out experiment to prove that DNA replication occurs according to semi-conservative model. Okay, so in order for these two scientists to prove that DNA replication occurs according to semi-conservative model, they breed or they culture like E. coli. Okay, they culture E. coli in two types of medium. The first medium, it contains the heavy nitrogen isotope, which is N15, and then after that, they transfer the bacteria into another medium which has the lighter isotope, which is nitrogen 14. So for the zero generation, basically, okay, zero generation, generation 0. Okay, so when the two scientists grows the bacteria in the medium that contains nitrogen 15, and then they centrifuge the DNA samples with cesium chloride, the cesium chloride will end up at the bottom of the centrifuge tube. Okay, so this is because cesium chloride has the same density as DNA. Okay, so when the DNA from the zero generation bacteria was centrifuged, they found out that this bacteria or this sample will produce a heavy DNA band, so it will be at the bottom of the tube. Okay, so this means that the DNA of the bacteria composed of the heavy nitrogen. Okay, this is 100%. Of the DNA has nitrogen 15. Nitrogen 15, nitrogen 15. After that, they transfer the bacteria in a medium that contains nitrogen 14. Okay, so this is generation 0. They waited for 20 minutes. Okay, that allows the bacteria to replicate, and then take the sample and then put it in the centrifuge tube and then centrifuge the DNA sample. So centrifuge is when you you put the DNA sample in the centrifuge machine and then you spin the sample at high speed. Okay, so this will separate the DNA into its density. So for the generation 1, they will use the template from the DNA template from generation 0 and used to synthesize the new DNA strand using nitrogen 14. Okay, so you have to understand, when you centrifuge the DNA sample, it produces only one DNA bands, okay, that have the intermediate density. Okay, the intermediate density. So this means that for generation 0, they will separate and becomes the template to synthesize the new DNA strand. Okay, so just imagine the red color here indicates that the DNA consists of nitrogen 15. So after they have transferred into nitrogen 14 medium, so the bacteria will use nitrogen 14 from the medium and synthesize the new DNA strand. Okay, so the blue strand here, it means that the DNA contains nitrogen 14. Okay, so this means that 100% of the DNA for generation 1 has intermediate density. It has both the old strand nitrogen 15 and the newly synthesized strand nitrogen 14. Okay, so this is, it produces DNA with intermediate density that consists of nitrogen 15 and 14. Okay, so that's why the answer is A. So this proves that DNA replication occurs according to semi-conservative model. But if you were to get the DNA sample for the second generation, okay, second generation, and you centrifuge the DNA sample again, so you will get two bands, two DNA bands. Okay, one of intermediate density, the other of light density. Okay, so this means that the DNA from the first generation will separate and then becomes the template to synthesize the new DNA in the second generation. So imagine you separate the DNA strand. Okay, the DNA strand. Okay, so and then use the nitrogen 14 to synthesize the new DNA strand. So the blue one indicates it is consists of nitrogen 14 only. So as you can see, 50% of the DNA consists of nitrogen 14, nitrogen 15. Okay, so this one is for the intermediate band. Okay, and then another 50%, it consists of nitrogen 50% nitrogen 14, nitrogen 14. Okay, so this is the light band at the top of the tube here. Okay, so this proves that DNA replication occurs according to semi-conservative model. Sickle cell anemia is a genetic disease caused by the replacement of glutamic acid by valine at specific position in the beta chains of hemoglobin. The valine makes the hemoglobin stick together, thus distort the shape of the red blood cells. So it means that the red blood cells cannot carry enough oxygen as the hemoglobin will stick together. As you know, the function of hemoglobin is to bind to oxygen. So when the hemoglobin sticks together, it cannot bind to oxygen, and so therefore red blood cells cannot carry oxygen. So the type of mutation that leads to this disease is basically, the answer is nucleotide pair substitution. So if you look at the question here, the key point you have to notice is here, the replacement of glutamic acid by valine. So this is when the mRNA codon, okay, that's supposed to code for glutamic acid is changed and changes to a different amino acid, which is valine. So if you look at what I've written here, the mRNA codons, these are the mRNA codon. Okay, so the mRNA codon for glutamic acid is supposed to read GAA or GAG if you refer to your mRNA codon table. Okay, so but if mutation were to occur and changes the second base of the codon from A to U, okay, so instead of coding for glutamic acid, it will change to valine. Okay, so for the codon for valine is GUA, okay, or GUG. So you can see the codon for the two amino acid are similar, but the second base is different. Okay, so so this will lead to the uh, the the change of amino acid, and this is actually a missense mutation. So the answer would be D, nucleotide pair substitution. It changes adenine to guanine. Okay, it is nucleotide pair substitution that will cause missense mutation, which is the codon will change from one amino acid to a different amino acid. That is the answer for question number 7, which is D. Which of the following is true about DNA transcription in prokaryotic cell? So the answer for A is obviously wrong. Okay, it is not correct because prokaryotes are bacterial cells. For example, E. coli. So for these prokaryotic cells, you have to look back at your chapter 2 last semester, which is cells. So for prokaryotic cells, they do not have membrane-bound organelles. Okay, so all their DNA composition is not within the nucleus. Okay, they are confined in a region, we call it as nucleoid. So A is wrong. Okay, B, the premature mRNA undergoes RNA processing from mature mRNA. So for statement B, prokaryotic cells does not undergo RNA processing. Okay, only eukaryotic cells undergo RNA processing that convert premature mRNA into mature mRNA. Okay, by, for example, removing um, the non-coding sequence, okay, and forming a continuous coding sequence. Okay, and then for B is not correct. Okay, statement B is not correct. For C, the mature mRNA is translated into to synthesize polypeptide chain in the cytoplasm. So again, this statement is not correct because for prokaryotic cells, it does not produce any premature mRNA. The information from the DNA, okay, is straight away used to synthesize mRNA, okay, during transcription, and then during translation, it is used to synthesize the polypeptide chain, okay, in the cytoplasm of the prokaryotes. Okay, so statement C is not correct. For statement D, a gene in the in the prokaryotic DNA provides the instruction for synthesizing an mRNA transcript. So this one is correct. So this is just a simple instruction or simple statement where the DNA from the prokaryote, okay, which is a segment of the DNA, which is a gene, is used to synthesize mRNA during transcription. Okay, so the statement D is correct. Question 9. What is the role of release factor during protein synthesis? Basically, during protein synthesis or translation, okay, at the end of translation, the termination step involves a release factor that enters into the A site of a large ribosomal subunit and breaks the bond between the last amino acid of the polypeptide chain with the tRNA that sits in the P site. So the statement D is correct. Okay, hydrolyzes the bond between the tRNA in the P site and the last amino acid of the completed polypeptide chain. So basically, you have to understand that during translation, the mRNA will be in between the small subunit and also the large subunit of ribosome. Okay, so ribosome is the organelles that involve in protein synthesis. So during the process of translation, the tRNA, okay, that holds or that binds uh, to the elongating polypeptide chain will sit in the P site. Okay, when in the A site is vacant, the next codon will be read. But if the codon reads for stop codon, so there are three possible stop codon that you have to remember, which is UAA, UAG, UGA. So if one of the stop codon enters into the A site, okay, so this indicates that termination will occur. So when a stop codon sits in the A site, so a release factor, okay, release factor will enter into the A site. Release factor is a type of proteins. Okay, so the function of this release factor is to break the bond, okay, or hydrolyze the bond between the last amino acid of the polypeptide chain and the tRNA that sits in the P site. Okay, peptidyl T site. All right, so that is the answer for question number 9. Question 10. Which statement is not true regarding the gene regulation of like operon of an E. coli? Not true. So first, before we go to the answers, you have to understand the concept or the structure of like operon and what is the function of like operon in E. coli. Basically, like operon is a portion of a portion of bacterial DNA, okay, that controls for the synthesis of enzymes, okay, that is used in the hydrolysis of lactose. Okay, so lactose, when it is hydrolyzed into glucose and galactose, then only the bacterial cell can use the glucose, for example, to undergo cellular respiration, okay, to generate energy for the bacterial cell. So you have to know what are the component of like operon. Like operon, like operon is consists of promoter, where RNA polymerase will bind to, promoter, where repressor protein will bind to in the absence of lactose, and then you have the three structural genes, which is like Z, like Y, and like A. From the three genes, okay, through transcription, it will produce a single continuous mRNA that is used to synthesize three types of enzyme. For like Z, it is coded to synthesize the enzyme beta galactosidase. The function of beta galactosidase is to hydrolyze lactose into glucose and galactose. The next structural gene is like Y. The function of like Y is to synthesize the enzyme lactose permease. The function of lactose permease is to allow lactose into the bacterial cell. Okay, enter into the bacterial cell, and the last enzyme, or sorry, the last gene is like A, which is used to synthesize the enzyme transacetylase. Okay, so ahead of the promoter, okay, ahead of the like operon, you have the regulatory gene. The function of this regulatory gene is to synthesize mRNA that synthesizes the repressor protein. In the absence of lactose, repressor protein will bind to the promoter and stop the transcription of mRNA for the 3 structural genes, and so therefore the 3 enzyme cannot be synthesized. Okay, so that is in the absence of lactose. In the presence of lactose, some of the lactose that enters into the bacterial cell will get converted into its isomer, which is allolactose. So allolactose then enters into the active site of or it binds to the repressor protein and causes the repressor protein to change shape. Okay, this is in the presence of lactose. Okay, so it changes shape and it cannot then bind to the operator. So once the operator is free, okay, there is no blockage. Okay, the promoter is not blocked. So RNA polymerase can bind to the promoter, and then from there the like operon is switched on and can synthesize the mRNA from the 3 structural gene to produce the 3 enzyme. So now let's look at the statement for the 4 possible answers. For statement A, okay, for statement A, the regulatory gene codes for the synthesis of repressor protein. So this statement is obviously correct. Okay, regulatory gene, which is this one, okay, codes for the synthesis of repressor protein. So that is not the answer. A is not the answer. So B, the structural genes are transcribed when the repressor protein binds to the allolactose. So as I said before, when lactose enters into the bacterial cell, some of the lactose will be converted into its isomer, which is allolactose. Allolactose will then bind to the repressor protein. Okay, repressor protein. So it binds to the repressor protein, changes the shape of the repressor protein, and it cannot bind to the promoter. It cannot bind to the operator. So when repressor cannot bind to the operator, RNA polymerase can bind to the promoter, and transcription of mRNA can occur to synthesize the 3 enzymes. Okay, so statement B is correct. So that is not the answer. The structural genes are transcribed when the repressor protein binds to allolactose. So this is in the presence of lactose. Like operon is switched on. Okay, so statement C. In the absence of lactose, repressor protein binds to operator region, and the like operon is switched off. So this is in the absence of lactose. When lactose is not present, repressor protein can bind to the operator and stop the transcription of mRNA. Okay, and then statement C is not it is correct, but it is not the answer. So statement D, the function of beta-galactosidase synthesized by like-Y gene is to hydrolyze lactose to glucose and galactose. So statement D is not correct. Okay, because beta-galactosidase is coded by the gene like Z. Okay, like Y is to synthesize lactose permease. So the answer will be D. D is not the correct statement. Okay, it is not true regarding like operon.